Olora Health, a team of biomedical engineers working to fill a gap in women's health. 30% of women worldwide will experience bacterial vaginosis in their lifetime, and up to 80% of these women will experience recurrence within just 1 year. That means millions of women are trapped in a cycle: infection, antibiotics, temporary relief, and then recurrence. The problem isn't just infection, it's recurrence. Current treatments rely on antibiotics that fail to eliminate bacterial biofilms and can disrupt the vaginal microbiome, making reinfection more likely. So, instead of restoring balance, treatment often sets the stage for the next infection. Our mission is to break this cycle and prevent recurrent BV, not just treat it.
We developed BVBalance, a soft, tampon-like insert used alongside antibiotics. Upon insertion, it provides immediate comfort through hyaluronic acid lubrication and expands to conform to the vaginal canal. Alongside a course of antibiotics, a biofilm-disrupting antimicrobial is released to enhance antibiotic effectiveness. After disruption, probiotic restoration begins during the critical post-treatment window. Finally, the device safely dissolves within 2 to 3 weeks, requiring no removal. BVBalance covers 3 critical areas that were defined through over 150 clinical and patient interviews to ensure we address real unmet needs. Unlike antibiotics, which relieve symptoms but fail to prevent recurrence, and unlike probiotics, which restore bacteria but do not address biofilm persistence, BVBalance combines both. First, combination therapy, disrupting biofilm and restoring microbiome balance. Second, sequential release. Restoration begins precisely when reinfection risk is highest. Third, preventative focused design, targeting recurrence, not just symptoms.
When compared to existing options, antibiotics, boric acid, and over-the-counter probiotics, BVBalance is the only solution that targets biofilm, restores microbiome balance, provides symptom relief, and is designed specifically to reduce recurrence. This positions BVBalance not as a replacement, but as a complementary advancement in BV care. Now, I'll walk you through our prototyping progress, where we've translated our concept into tangible designs and early validation. We're advancing 4 key areas in parallel. First, our technical model is guiding the design of the device. These models simulate how our antimicrobial is released over time and how different scaffold geometries impact that release, allowing us to optimize for sustained concentrations needed to disrupt BV-associated biofilms. Second, in collaboration with Mayo Clinic, we've initiated in vitro biofilm studies using Gardnerella vaginalis. Early results show our antimicrobial approach is effective in reducing biofilm viability, supporting our therapeutic strategy. In parallel, we've begun PLGA sheet formation using solvent casting. This gives us a controllable platform to evaluate material properties and validate release behavior against our model predictions. Third, we're developing a 3D printed scaffold, where we're tuning lattice structure and porosity to balance mechanical integrity with effective drug delivery. Additionally, we are designing a user-friendly box that will house a device, ensure proper storage, and support ease of use.
Our value proposition is simple. It targets the protective biofilm layer of G. vaginalis, which allows the current antibiotic to be more effective, reducing recurrence rates, improving treatment outcomes, and lowering long-term healthcare costs. Key stakeholders include gynecologists and primary care physicians who treat BV, treatment-seeking patients, pharmaceutical partners, and healthcare systems that focus on recurring infections. The market strategy begins with positioning the product as an over-the-counter adjunct therapy that may be prescribed alongside metronidazole, targeting a large population of women experiencing recurrent BV. Ultimately, we want an effective, microbiome-friendly treatment that addresses the root cause of reoccurrence.
To move beyond proof of concept, we are seeking $100,000 in funding, along with strategic partnerships. This funding will support expanded antimicrobial and biofilm testing to validate our results across a larger sample set and optimize selective activity against pathogenic bacteria while preserving beneficial microbiota. We also plan to pursue early FDA engagement through the INTERACT pathway to align on our regulatory strategy and reduce development risk, as well as secure patent and legal protection for our formulation and delivery approach. In parallel, we are seeking partnerships in microbiology and regulatory advising to strengthen our development pathway and support the transition from early validation to a clinically and commercially viable product. Thank you.